Welcome to Life and Biology. This is Dr. Joel Graff, and I'm going to be discussing some overhead slides that I drew on a lecture given on February 9th, 2022. And we did a quick review of the last thing we ended up talking about on the previous lecture, and that was enzymes with and without cofactors. And so an enzyme without a cofactor is called an apoenzyme. Enzymes with a cofactor are called holoenzymes. I want to be clear that not all enzymes require a cofactor, but the ones that do, it's important because without the cofactor, an enzyme's not very efficient. With the cofactor, the enzyme can be more efficient. So the cofactor will either be removed from the protein or else they can be added in. And so that's we've got the cofactor here. Now a cofactor, examples of cofactors might be calcium, zinc, magnesium. So there's different things that enzymes can bind to. And cofactors are usually an ion of metal. Now, there's a second type of thing that can bind to enzymes to make them holoenzymes, and that would be called a coenzyme. Now, a coenzyme is also a cofactor. It's just all coenzymes are cofactors, but not all cofactors are coenzymes. And in biology, these cofactors that are coenzymes often take the form of a vitamin. So if you are eating your box of cereal in the morning, the food label information, the large print is going to be dealing with macromolecules, the ones that are giving you the calories, your fats and carbs and protein. And then in fine print, they're going to be telling you about things that you don't need as much in your diet, but there is recommended amounts that you should be getting. And the reason there's recommended amounts is because if you don't eat food with enough minerals and vitamins, then you could end up having a situation where there's not enough of those cofactors for the enzymes. And if you don't have cofactors for those enzymes, then your cells aren't going to work properly because some of their metabolic pathways aren't going to work properly. So that is why you need some minerals and ions in your minerals and vitamins in your food to be healthy. It will make all these enzymes run more efficiently. Of course, things like calcium are important for making bone and other things, but there's more than one reason you might need some calcium. But anyway, when you read about a lot of those, they're because they're cofactors for enzymes. Okay, now we're working our way up towards metabolic pathways. That's when you take one type of molecule, act on it with an enzyme, and then you can have a new molecule, and then it acts on and it can get acted on again by another enzyme, and you have these chains of input-output reactions. Now, cellular respiration will be the subject, and the overall formula for cellular respiration is C6. This is hard to read, but it's H12O6. And then if that is combined with 6 oxygen molecules, those are the substrates going into cellular respiration. Now there's a whole lot going on with this arrow, and we're going to talk about all the different steps involved, but in the end you end up with 6 carbon dioxide molecules and 6 water molecules. Okay, so this is the balanced reaction for cellular respiration. Now sugar has a lot of energy in it, and so the sugar and oxygen together have more energy than the carbon dioxide and the water that you end up with at the end. And so because we're going to be going from high energy to low energy, this is where we're going to have some energy released. And if you are doing cellular respiration, some of that energy will be released as heat. But if you're doing cellular respiration correctly and making use of the energy that was in the sugar, now remember the sugar is a molecule, so it's chemical energy. You could also trap that as chemical energy in the form of ATP molecules. So that's the point of cellular respiration, is to break down this high energy sugar, collect as much as we can in the form of ATP before we end up with CO2 and water at the end. Okay. just like most reactions that are going to be from a high energy level to a low energy level for the substrates to be high and the products to be low, you're going to have a downhill slope, and you better hope that there is some activation energy slowdowns on the way, and that because if you just took the sugar and went straight to CO2 and water by having some oxygen, that is the same formula as fire. So if we burned something made of a bunch of sugar molecules like a log in our fireplace, all that's going to be released as heat. Now we don't want to catch on fire, so we're going to break down the process of collecting the energy out of the sugar into lots of little steps so that it's regulated and doesn't go too fast. Okay, and then the neat thing about photosynthesis is that if we look at the overall formula for photosynthesis, you have 6 carbon dioxide, 6 water, and they go to form a sugar and 6 oxygen. And so that is the exact reverse reaction of cellular respiration. This is also a multi-step process, and so we're going to go from low energy carbon dioxide and water as the substrates of the reaction to a high energy sugar, and oxygen's a byproduct. And so we need to add energy into the system as we're doing that, and that energy comes from the sun. And so the energy from the light, typically the sun, but I suppose it could be a grow light indoors, is going to be captured within the process of photosynthesis, and the outcome will be a high energy sugar molecule. Okay. Now, excuse me. Enzymes can be inhibited, and so this is a little out of order. I suppose this would have gone better right after the cofactors since we were talking about energy regulation. But anyway, here we're on to energy or enzyme inhibition. In the case of enzyme inhibition, there is a certain type of inhibitors called competitive inhibitors. And in this case, your substrate might be kind of a triangle shape like this to fit in the Pac-Man mouth, the active site. Oh, and that's spelled wrong. It's S-I-T-E. I guess I was just used to putting an I on Pac-Man, so I was thinking site, the wrong kind of site. Anyway, a substrate can enter into the active site, but if there's another molecule called an inhibitor around that is very similar in shape and structure to the substrate, it could also end up sitting in the active site. So you have a competition. Who's going to get to the active site first? And if it's the inhibitor, then the enzyme can't work on the substrate if an inhibitor is in its mouth. Then we have 2 types of inhibitors that both have this word steric in them. One is just straight up steric. The other is allosteric. Allosteric means without, or and then steric. So what does steric mean? Well, steric is usually used like stereo or 3D, taking up 3D space. Was allosteric is not taking up the space, not competing for the same area. Okay, so in a steric inhibitor, the inhibitor binds not inside the active site, but it binds in such a way that it's blocking the active site. And if we had this substrate go down and try to go in there, if I had drawn correctly, this inhibitor would be blocking the active site so that the substrate couldn't make it in. The allosteric, that's without actually blocking the active site. These can work in 2 ways. What it means is the inhibitor will bind to the protein somewhere other than the active site, or somewhere other than an area where it would block the active site. And what it can do is, one, it could change the shape of the enzyme. So if it binds here, maybe the enzyme's active site becomes smaller, and now you can't fit the substrate in the active site. The food is too big for the mouth. Or if the inhibitor binds to the protein, it could inhibit the protein from being able to change shape. And if it can't change shape, then it wouldn't be able to do the induced fit. And so then it wouldn't be able to, once the substrate was in its mouth, do the clamping down on the substrate. So a couple different ways that an allosteric inhibitor can work. Again, the first way was that it changes the shape so that then the substrate can't fit in its mouth. The other way is if it binds to and prevents the movement of proteins. So, and that brings up an important point. Proteins are often drawn as like a static image, but in reality, they have movement to them. And so, remember all that when you're thinking about proteins. Okay, now we're back to metabolic pathways. So here I drew A through D in capital letters, and these are going to be metabolites. So you have a molecule called A, so it's metabolite A, another molecule called B, molecule called C, and molecule called D. So molecules that are involved in a metabolic pathway are metabolites. Then the metabolic pathways have enzymes that can help speed up reactions, and metabolic pathways can be regulated through feedback mechanisms, either positive feedback or negative feedback. So in this case, I drew A, B, C, and D. We have an enzyme called AB that will clue you in that it works on A to make B. Enzyme BC takes metabolite B and converts it to C. And enzyme CD takes metabolite C and converts it to D. These aren't real names of enzymes, but kind of like how lactose is broken down with lactase and sucrose is broken down with sucrase, sometimes the name of the enzyme can clue you in as to what they do. So that's why I gave them these particular fictitious names so that we could remember where they are in this pathway. Now, feedback loops can be either positive feedback or negative feedback. In this metabolic pathway, hypothetical metabolic pathway that I drew, I was saying that maybe if there's a lot of, maybe if you get the molecule called C built up, it could potentially go back and speed up enzyme AB. And so then you're speeding up the rate at which C is getting made. Then you could have an enzyme CD that converts C to D, and when the C gets converted to D, the molecule D could actually go back and be a negative regulator of some enzyme in the enzyme chain. And in this case, I drew it to enzyme B and C. So in one case, we've got a hurry up and make as much C as you can. And then we've got another case, or in the other case, as C is getting converted to D, it's telling the system to slow down converting B to C because it's already got enough D. So there's all sorts of interesting pathways like this in metabolism. The feedback regulation is where you actually get interesting, and it can form a system and a regulated system. So we will talk about when we get to cell respiration and photosynthesis, we're going to mostly be talking about molecules going from A to B to C to D, but if we had more time in the class, we would talk about all of the feedback that is built into those metabolic pathways, and then it gets really interesting. But we don't have time for all that. Speaking of, we're going to switch gears, and in the class, I showed this figure. And I don't know if this figure's copyrighted, but I imagine some of the figures that I showed were copyrighted. So I'm not going to go past this figure. But what we did in class is we brought up the idea of the phylogenetic tree of life. In that, we have a last universal common ancestor. So this is the base of the family tree, basically, family tree of life. And at some point, you had a diversion. The family's separated, and some of the living things were different enough from the others to be considered different branches on the tree of life. The first things we had were bacteria and archaea, and then later on, archaea, one of the archaea cells ate a bacteria cell, and that was the birth of the eukaryotic branch. And so in the first day of class, I had it to where the archaea and bacteria branches went together to form the eukaryotic branch of life. But there's different ways of showing this. Mostly what I wanted to bring up is the idea that when I was a kid, we learned that there were 5 kingdoms of life. There was the prokaryotes or bacteria, there was protists, which are a lot of the ones shown down here. There were plants, fungi, and animals. So 4 of the kingdoms of life were actually eukaryotes. And the final kingdom of life, which we called bacteria at the time, turns out they were actually 2 different things. So it wasn't until we were able to sequence bacteria and archaea that we saw that they're actually different, completely different organisms, and they were very evolutionarily distantly related. And so just by looking at it, you can't tell really the difference between a bacteria and archaea. So anyway, so what I did for the rest of the class, and I'm just going to skip it here, and you can, if you're in my class, you can go look at the class notes, but I walked through a bunch of images of bacteria, pointing out all the different features you might find on a bacteria. Then I showed an archaea, which looks very much like a bacteria and had basically all the features we'd already talked about for bacteria. And then we got to the eukaryotic branch of the tree of life, and I walked through a bunch of the examples of cells from the eukaryotic branch, because in eukaryotes you have some single-celled organisms, multi-celled organisms, cells that have plant or cell walls like plants and fungi, and cells that don't have cell walls like animal cells and flagellates and diplomonads. But anyway, so that's what the rest of the lecture was, is that I just kind of walked through pictures. So on your own time, just if you're in my class, go ahead and walk through those pictures. If you're not in my class, go ahead and take some time to go do some Google searches on the internet for examples of cells of these different kinds. Diplomonads is Giardia was an example. Flagellates, I gave the example of Leishmania, which is a parasite that I studied back in the day. It causes a disease in humans called leishmaniasis. And then fungi, you can have single-celled fungi, which are called yeast, or multicellular filamentous young fungi. So go ahead and take a minute to go look at some of these different cell types. Anyway, this has been Life in Biology. I'm Dr. Joel Graff, and you can like and subscribe or not.